inviting me. So I'm from University of East London. I'm a senior researcher there, but I am the CEO of ZEAB Therapeutic in United Kingdom. So we are a life science research company and recently opened our diagnostic lab in March this year. So this presentation is one of the research, work project that we are working on, and I'll be talking on exploring and exploiting the homeostasis network, orchestration, and biomarkers in human health disease prevention. And I think this topic is really important to this conference because it will really help to inform on involvement in primary healthcare and Medicare. So like I mentioned, we do have conflict of interest is one of our projects at ZEAB Therapeutic Limited, so, and I'm the CEO. Overview of my talk will involve a brief introduction of what homeostasis is, the aims and objective of the project, the research method that was used to conduct this project, the results, and summary and conclusion. So for introduction, we all know that every human body undergoes survival techniques, and one of that survival technique is to maintain an internal environment. So also a disease state, for a disease state to progress, there has to be an internal environment that disease state must maintain, such as cancer. So cancer cells, they maintain internal environment within an allowable limits that enables their survival, as well as promotes the proliferation of that cancer cell. So my main topic will be regarding colorectal cancer, because that's what I major on. And looking at colorectal cancer, we know that aside from this recent pandemic, that COVID-19 is a major killer worldwide now. From this has been from 2019 December up to this moment. So the global scientific body has been putting in so much effort in looking into resolving this pandemic of COVID-19. However, colorectal cancer is also seen as a major concern because it is epidemiological report says that is the among women is the second most mortality causing disease, while among men is among women is the third, and among women is the second mortality causing disease. So colorectal cancer is associated with perturbations in cellular metabolism and increase in glycolytic flux. So in this study, we were able to look at the importance of homeostasis level and how this homeostasis, once it's perturbed, how does this facilitate promotion of the colorectal cells. So we all know that we looked at bile acid, using bile acid as an example, because bile acid is a byproduct of steroids and a metabolite of steroids. So this project was actually conducted with ZEAB Therapeutic alongside Human Metabolome Technologies company. They are based in United States in Boston. So they supported this project and did the analysis of the samples. And we used targeted quantitative metabolic analysis performed on colon cancer cells as well as normal cells. So, and the colon cells the colon cancer cells that were used were HCT116 and HT29, and the normal cells were CRL1790, which were purchased from ATCC company. So a total of 116 metabolites were looked at, but I'm not going to present all the data in this talk because of time. So I'm only going to look at few parameters that we looked at, up to 3, that because of the time frame for this presentation. So the results were measured in terms of picomoles per million cells. So in summary, of what I'm going to be talking about is that we're going to look at metabolites, we're going to look at the ATP energy metabolism, we're going to look at the GSH and GSSG ratio, which tells us about antioxidative ROS response, that reactive oxygen species, and we're going to look at betaine, which is a response to osmotic pressure and pro-inflammatory. So this data, these are the data, and this data looked at the NAD ratio with NADH, and which is a ratio of glycolytic flux or the Weibuck effect. We can see in these data, I hope you can still see my screen. So we can see in this data that—
Yes, we can.
Yes, we can see in this data that deoxycholic acid, which serves as a tumor-promoting substance, really caused a lot of perturбation of the parameters that we are looking at. And we noticed that in normal cells, this bile acids, deoxycholic acid that is indicated as DCA here, was able to disrupt the normal states, the survival states of the normal cell, and in comparison to the colorectal cancer cells, the HCT11Cs and HT29, we noticed that aside from the fact that you have that these two cancer cells are cancer cells that are already in a disease form, the presence of deoxycholic acid still, enhanced perturbation of the states of these cells, thus even promoting proliferation and causing further, should I use the word, virulent expression of the morphology of the cells. The morphology picture is not shown here, but we observed that there was a remarkable effect of this of the deoxycholic acid on the morphology of these cancer cells, despite they're already cancer cells, so causing further damage to the cells and making them even more aggressive cells. So this is continuation of the glycolytic measures that were observed, and we noticed that there was so much perturbation in these parameters in relation to, in comparison to normal cells. Also, I'm sorry if I'm too fast, just slow me down. I'm just trying to talk within my time frame that was given to me. So we also looked at ATP level in the cells. So we observed that we all know that glycolysis provides ATP by substrate level phosphorylation, as well as reducing equivalent NADPH. So we looked at the ATP parameters and observed that with the deoxycholic acid, which is a tumor-promoting substance, also cause a perturbation of these parameters in the normal cells. And in the presence of bile acid, this perturbation was reduced. Also, additionally, we looked at another type of bile acid, which for the sake of confidentiality will not be mentioned in this presentation, and is indicated as R, as you can observe in my data, that this bile acid was able to negate the effect of the tumor-promoting substance, that's deoxycholic acid. Thus, making us showing us the importance that an understanding of the homeostasis level in any individual is very important. And once this homeostasis level is perturbed, this is what brings about promotion of initiation and promotion of disease, in this case, colorectal cancer cells. This is a continuation of the previous slide on ATP. We can see that with the with the dietary fiber and the R bile acid that is indicated here, we noticed that the effect of deoxycholic acid was reduced in the presence of the bile acid and the R bile acid. So we also looked at another parameter, which is betaine, and betaine actually, causes inflammation, inhibition of cell growth in this cancer cells, because with these parameters, what we were able to notice was that there was a kind of a network, a connectivity. And what we meant by this connectivity, I will explain it in the next slide, was that we noticed that as you observe the increase in ATP production, as well as the NADP, the first data slide that we observed with betaine, you can see that there is what we call orchestration of parameters among this. So we observed that bile acid and the R indicated by, we observed that the dietary fiber and the R bile acid were able to kind of stabilize the homeostasis level in normal cells when the deoxycholic acid perturbed the homeostasis level. So we observed that with bile acid, the effect of, we observed that with the dietary fiber, the effects of the damage caused by the deoxycholic acid was observed more with the presence of the R bile acid, meaning that the dietary fiber worked better with this R bile acid when deoxycholic acid perturbs the system. So this was observed in the GSH and the GSSG ratios. So glutathione is an important element in homeostatic system, and with this GSH, we linked our GSH parameters with the other parameters that we have observed. And then on the next slide, I'm going to explain further why we had to link this and let you know more on the network that we are talking about. So on this slide is, we did a qPCR analysis of GST-M1 and GST-T1 polymorphism. So GST-M1 and GST-T1, these are a family of enzymes that are responsible for the function of glutathione. They bring about the conjugation of glutathione for its effectiveness in its function in homeostatic system. So our data show that with the colorectal cancer cells, though the effect of deoxycholic acid that was seen in them and the effect of dietary fiber and the R bile acid that was seen in these cells seems to be similar, these effects were not duplicated, because these two cancer cells have their different mutations, their different level and stage of disease and type of colorectal cancer. First, we noticed that the GST-M1 and GST-T1 is present in HT29, but not in HCT116. So this kind of informed on why we noticed the difference in the effects of treatment that were observed on these two cells. So in summary, I hope I'm still within the time frame. So in summary, we noticed that both HCT116 and HT29 exhibited treatment effects from fiber and bile acids. DCA, which is deoxycholic acid alone, appeared to increase oxidative stress, as mentioned earlier, that this is a tumor-promoting substance, it's a tumor-promoting bile acid. So it was able to cause oxidative stress, osmotic stress, drive the ATP demand, and increase NAD and NADH ratio in the cells. And with R, we alone, these effects were that was caused by deoxycholic acid was inhibited once every, that's the effect that deoxycholic acid caused was inhibited by the R bile acid, so thus decreasing the level of oxidative and osmotic stress and energy demand. So this, in healthy cells, there were likely unaltered effect of dietary fiber and the R bile acid, but deoxycholic acid was able to cause changes in the normal cells because it's a tumor promoting cells, as mentioned. So with the presence of dietary fiber and the R indicated bile acids, we observed that the effect of deoxycholic acid was inhibited in the normal cells. So these effects were similar between HCT116, but not duplicative, because these two cells are different cell lines. So in conclusion, bile acids, has been shown by various literature and study, that bile acid induce cancer in rodents, and precisely the secondary bile acid, deoxycholic acid, has been a major culprit of this. And research has shown that dietary fiber is important in reducing the effect of bile acids. Also, what this study is actually showing is that with paying attention to homeostatics in individuals, paying attention to the homeostatic system, we will be able to monitor and manage diseases more appropriately, and even giving precision to patients, because individuals have different, should I use the word, polymorphisms or different mutations in their homeostatic systems. So this work has been made possible with the support from HMT, particular gratitude to Dr. Alice Bucco, who is the Vice President of HMT. Then I have the staff that worked on this study was Dr. Oyi Udo Chijoke and Dr. John Patrick Alao. So both are staff of ZEAB Therapeutic that worked on this study. And we have consultants that also worked with us from Nigeria. We have Dr. Solomon Rotimi, we have Ademola from University of Ilorin, and we have Professor Winston Morgan at University of East London, which was the base lab where this work was carried out. Thank you for listening, for your audience, and I hope you do have questions for me, and if not, you can always reach me via email.